Welcome to this video abstract. I'm Christina Tremblay, and I share the first authorship with Dr. Abbasi on the article accepted for publication in Brain, titled Sex Effects on Brain Structure in the Novo Parkinson Disease, a multi-modal neuroimaging study. The paper discussed work conducted under the supervision of Dr. Alain Daguerre at the Montreal Neurological Institute, affiliated with McGill University. Parkinson's disease is associated with clinical symptoms varying in their occurrence and severity, and sex may play a role in this heterogeneity. For example, the incidence of Parkinson's disease is twice as high in men than women, and men tend to have an earlier age of onset. As the disease progresses, women suffer more from anxiety and depression, but men show more cognitive deficits. Regarding motor symptoms, women show more tremor, while men generally present more rigidity or bradykinesia, indicative of a more severe phenotype in men. This is in line with studies demonstrating a more benign phenotype in women and a neuroprotective effect of estrogen. Studies on animal models have revealed that estradiol administration is associated with decreased dopaminergic neurodegeneration and increased dopamine receptors in the striatum. In humans, estrogen depletion in different conditions, including ovariectomy, has been shown to increase the risk of developing Parkinson's disease. In addition, beneficial effect of estrogen use was observed on symptom severity at the early stage of the disease. These findings suggest that men with Parkinson's disease may have greater neurodegeneration than women. However, little is known on sex differences in the pattern of neurodegeneration. In this paper, we aim to compare structural brain features in men and women with Parkinson's disease using multiple neuroimaging measures such as deformation-based morphometry, DBM, cortical thickness, and division-weighted imaging in a large sample of patients. We use MRI and clinical information of 149 men and 83 women with de novo Parkinson's disease from the PPMI database. The men and women groups had comparable clinical severity as shown in the table. We also use the information of the healthy control from the same database to calculate W-score maps to regress out the expected effect of age and sex for each neuroimaging measure. I will present our findings in the next slide. The DBM W-score maps were used to compare the mean regional deformation of the brain between men and women. Men with Parkinson's disease had more atrophy than women in 11 cortical regions, in blue here, and the left thalamus, while women had greater atrophy in 6 cortical regions, in pink. Using diffusion imaging, we observed more disruption in local efficiency of the structural connection in men compared to women, in multiple cortical and subcortical regions, such as basal ganglia, hippocampus, and thalamus. But such difference between sex was not observed in cortical thickness. Even though men and women with Parkinson's disease had similar clinical disease severity, this study showed that neurodegeneration and white matter damage are already more severe in men at the earliest symptomatic stage. Estrogen neuroprotective effect could potentially explain why men with Parkinson's disease had more atrophy and widespread white matter damage. The distinctive phenotype and brain differences between sexes support the idea that the development of Parkinson's disease may involve different pathological mechanisms and yield distinct prognosis between men and women. Thank you for listening and to all the funding agencies. For further discussion, please reach us at the following email addresses.